Hello, everyone, and welcome to the third and final installment of UHN Foundation's three-part Krembil Discovery live stream series. Today, we shine the spotlight on the Donald K. Johnson Eye Institute, Canada's largest research and eye care program. I'm Christian Cote, host of Behind the Breakthrough, the podcast all about groundbreaking medical research and the people behind it at the University Health Network. We hope you are staying safe and well and taking care of each other as we begin to see the light at the end of the tunnel of this COVID-19 pandemic. And as valued members of our community, we are so pleased to be bringing you this exclusive opportunity and up-close access to the amazing scientists and their innovations that are ongoing at the UHN every day. A little bit of background for you about the Krembil Discovery live stream series. It was inspired by the generous donors to the 2020 Discovery Ball. That was, of course, meant to be an in-person celebratory event, but public health and safety guidelines required we postpone. There is thankfully a silver lining here, and that is in an incredible show of support, our sponsors and donors stepped up to honor their donation commitments. So that gesture of is testimony to their unwavering belief in the research ongoing at the Krembil Research Institute. And thanks to their generosity, what that means is the funds will support a portion of the Krembil Research Institute core budget, which supports all three of the institutes we've been highlighting in this live stream series. Today, we're talking one-on-one with the co-director of the Donald K. Johnson Eye Institute, Dr. Valerie Wallace, about her vision for the Institute, as well as her own pioneering research. We're also going to introduce you to some of the rising stars at the Donald K. Johnson Eye Institute, and we want to get you involved in today's conversation. So as the live stream unfolds, if a question comes to mind, please send it in. And what you do is you go to the link at the bottom of your screen or go to slido.com and type in code, so that's CODE, code 5875 to submit a question, and you can also upvote a question you'd like to hear answered. We'll get to those a little later in the live stream. Just a reminder, please, we can't answer any questions of a personal medical nature. Now, for those of you who may be unfamiliar with the three pillars of research being generated at the Krembil Research Institute, our communications team at UHN Foundation has put together a short sizzle reel to bring you up to speed.
Diseases of the brain, joints, and eyes impact millions around the world, more of us every day. Most medications and treatments address symptoms. What we need are cures. The brain is the most complex and least understood organ in our body. Researchers at the Krembil Brain Institute are not only unraveling its mysteries, they're on the cusp of game-changing breakthroughs to halt the progression of dementia and Alzheimer's, to treat complex brain tumors, stop seizures, diagnose Parkinson's and other movement disorders earlier and with more accuracy, to help people walk again, stop stroke damage, and even prevent strokes from happening. In other words, they're using their brains to save yours. By the time arthritis is diagnosed, the damage is often done. The Schroeder Arthritis Institute is decoding the complexities of arthritis to build a cure. Here, researchers are developing blood tests to diagnose it early, predicting outcomes of surgical and medical treatments to provide ultra-precise and personalized care, and working on world-first therapies to treat, halt, and even reverse the damage. Their ultimate mission? To cure arthritis in our lifetime. Diseases of the eye impact people of all ages and stages, and they're on the rise. The Donald K. Johnson Eye Institute is gaining new and extraordinary insights into these diseases to slow vision loss and help people see again. They're working to restore eyesight through cell transplantation, using 3D printing and developing a drug to treat glaucoma. They're exploring how virtual reality can help retrain the eyes of people with vision loss, and working on a drug that could save eye cells from dying. It's not just about giving people back their sight, it's about giving them back their lives too. For researchers at UHN's Toronto Western Hospital, relentless is more than just a word. It's a driving force. After all, we're not just here to study these diseases, we're here to stop them cold.
So clearly, the relentless pursuit of new treatments and cures is in the DNA of all the scientists at the Krembil Research Institute. And critical to harnessing and nurturing that talent is leadership. And that's where we turn to Dr. Dawn Weaver. Dr. Weaver is the director of the Krembil Research Institute and research co-director of the Krembil Brain Institute. Dr. Weaver is an award-winning scientist and a world-leading researcher in the search for a cure to Alzheimer's and dementia. Welcome, Dr. Weaver.
Thank you, Christian, and welcome to all our viewers. Thank you ever so much for joining us on this beautiful, nice summer afternoon. So as our viewers are aware, as you are aware, University Health Network is the largest hospital in Canada. It's a very complicated place. I like to say that UHN consists of the Toronto Western Hospital and several other less well-known institutions on University Avenue by the names of Princess Margaret and Toronto General. And every one of these sites and hospitals within UHN has a research institute associated with it. And the Krembil Research Institute is that institute which is associated with the Toronto Western Hospital. And what is it that we do at the Krembil Research Institute? We have three areas of focus: vision and disorders of vision, brain, disorders of brain, and arthritis and disorders of joints. And when you first think about it, you go, well, those are three very different areas. And they're not really. If you look at why people end up in long-term care facilities, those are the three leading reasons. Perhaps they're blind or have visual disorders such that they're no longer able to care for themselves. They may have disorders of the brain, stroke, dementia, and so that they need care. Or finally, they may be sufficiently affected by arthritis that they just can't manage at home alone. And so these three broad categories are why people end up in long-term care facilities. And at the Krembil Research Institute and at the various sub-institutes that compose us, we are absolutely dedicated to improving the quality of life and ensuring that people have a good life at home as long as possible. And it's because of these three areas that we are focused with three institutes. And today we are focused on the Donald K. Johnson Eye Institute. And you're going to be hearing a lot of the exciting research that we have ongoing here. And it is exciting. They're doing some really nice stuff, which I think is you'll be absolutely fascinated by. Before I turn it back over to Christian to get things going, I just want to acknowledge how important your support is to us. Certainly funding from the Krembil Foundation and the Krembil family and funding from Donald K. Johnson have been absolutely essential to the creation of the Krembil Research Institute and the Donald K. Johnson Eye Institute. And we are thankful for all of our donors who make the groundbreaking research that we do, and just a fraction of which you're going to be hearing about today, possible. So thank you and enjoy the next hour.
Thanks so much, Dr. Weaver. And I'm hoping you can stick around. I want to bring you and Dr. Wallace back into the conversation a little later in the hour. First, though, I do want to bring in Dr. Valerie Wallace. She is co-director of the Donald K. Johnson Eye Institute. She holds the Donald K. Johnson Chair in Vision Research. She is a senior scientist at the Krembil Research Institute. She holds a Tier 1 Canada Research Chair in Retina Regeneration, a career scientist in the study of neurodevelopment, degeneration, and regenerative medicine. Dr. Wallace heads up an innovative and world-leading research program with three primary goals: understand what causes vision loss and blindness, discover how to diagnose eye disease early, and finally develop therapies to preserve and restore sight. Welcome, Dr. Wallace.
Hi, Christian. It's great to be here, and thanks to all of you for joining us this afternoon.
Let's start big picture. What are your current priorities for the Donald K. Johnson Eye Institute?
So, I'd like to say we want to recover from the COVID disruption, which hasn't all been bad, and we have a number of things to capitalize on. It was a good time to reflect on research priorities, to do the groundwork about developing new initiatives. And we made some really big progress in the last year, including 2 recruits. Dr. Karun Singh, who you'll hear from later today, joined us last summer in the midst of all the COVID crazy. And Dr. Brian Ballios will be joining us in the next few weeks, our new clinician scientist recruit. So I think, we've hired new trainees as well, and we have a lot of very interesting nascent collaborations to kickstart. So I'm really excited about what we're doing right now.
And Valerie, can you give us a sense of, the next few years? Like, where do you want to take this place? What's the lighthouse in the distance that you're guiding everyone towards?
Absolutely. So at the core of this is, a lot of these age-related blindness conditions. The reason we don't have a lot of treatments to restore vision is because ultimately we really don't have a handle on the nuts and bolts of those diseases. Why do they happen anyway? And so I think over the next few years, through philanthropic support, we've been able to grow a critical mass of basic researchers, provide cutting edge resources, bring on new technologies and model systems. And ultimately, I think what I'd like to see and what will happen is turn these very smart people, turn them loose, set them loose on this problem, and forge even stronger links with our clinician investigators and the clinical group and get at disease mechanism. And that's the only way, I believe, and this is the tried and true thing, if you put smart people in a room, let them loose, they will have an impact. Can I tell you what that's going to look like specifically? No, but I don't think I need to. I think we will get there, and we already have discoveries in the last few years that show us that we're on the right track.
All right, let's turn to your research. And first, an understanding, maybe we'll step back for a second, understand the stakes involved, because you hinted at it a bit in terms of eye disease, and this is an aging disease. Speak to the scope of eye disease in Canada and the scale of how it affects quality of life for so many Canadians.
Sure. Well, so if you look at the most recent stats, right now it's estimated that 1.5 million people in Canada are blind or living with significant visual impairment. And that number is just going to grow because a lot of the diseases that are associated with these conditions are diseases of aging, and our population's aging. And the big players would be diabetic retinopathy, glaucoma, macular degeneration. So if I could give you some numbers, they estimate that 30% of people, there's a 30% risk of developing a macular degeneration by the time you're in your 75th year. So the costs to the healthcare system are staggering, $8 billion, dealing with people with this kind of vision loss. And that's just the healthcare piece, but not to mention the loss of quality of life and independence for people who have the conditions and also for their families. So not everybody who gets old gets these diseases, but aging is a big risk factor for getting these diseases. So if we could turn that around, I think we could have a really big impact on improving the quality of life of older Canadians.
I understand, Dr. Wallace, that the premise of your research is to inject stem cells into the damaged retinas of say someone with macular degeneration, and specifically, you're trying to replace the cone cells on the retina that have been damaged. Why the cone cells?
Okay, well, the way your eye works is it has to capture light to convert it to electrical signals. And that's done by 2 classes of photoreceptors, rods and cones. These are the light sensitive cells that line the back of your eye. The cone cells are what you need to see detail, faces, color, what we call high acuity vision. The vision that you probably rely on just to use this screen this evening. And those cells are lost or damaged in conditions like macular degeneration. So therapeutically, they're a really important problem in the clinic because there's a lot of people who are going to develop macular degeneration. If those cells die, they aren't replaced by any natural means by the body. So our idea is transplant healthy cells, and maybe they will, plug into the retina and restore light sensitivity to the retina so that now people could have better reading ability, better ability to navigate their world. So that's why we were doing cell replacement with cone photoreceptors.
Okay, so let's get a snapshot of what's actually happening in your lab. I know you work with animal models. Talk to us about what you've discovered so far in terms of once the stem cells are transplanted, how are they performing once they arrive at the damaged retina?
So absolutely. So most people, including myself, thought about this as a cell replacement. So we're adding healthy new cells and they're just going to set up shop and replace the lost ones. That's what we thought would happen. And what we've discovered is, no, they do something very different. They actually engage in a kind of handshake with the cells in the recipient retina where we transplanted them. And instead of replacing lost cells, what they do is they attach to the remaining cells and they transfer material. So we see that as transferring, the fluorescent proteins that we use to tag them. Those are engineered things, but we also know they transfer normal things that they make. So that's a very different way of thinking about how cells might restore function. So one possibility is that this exchange, this kind of handshake, could help improve the function of kind of sick or struggling cells in a diseased eye. But what it's also shown us is that when, for example, in the case where we've lost all those cells, so the host retina that doesn't have any of these cells to make the handshake with, that we really need to understand how to get these cells to plug in and restore vision. So actually function as cell replacement. So we're really interested in looking at those two ways that transplanted cells could restore vision in a damaged eye.
Okay, so where are you at then in terms of that?
Where are we at?
That goal of trying to restore vision?
Well, so we're now testing whether this handshake mechanism can help restore vision in some circumstances. And if we understand that, maybe we could exploit that in some types of retinal disease. We're also working very hard to understand how photoreceptors, even healthy ones in a normal retina, actually form those synapses, those plug in and talk to the retina kind of communication, the way they organize to communicate. So we're trying to understand that in how this is happening normally, but also transfer that information to coax, transplanted cells to do that, to engage in that behavior. So that's where we are right now.
And I don't know if you could blue sky, say 3 to 5 years from now, what would be your, a best case scenario for your lab?
The best case scenario for my lab would be that, for example, that we would have evidence that, for example, maybe we can manipulate transplanted cells and coax them into forming synapses and show that that gives you an even greater boost in vision rescue after transplantation. And then maybe those strategies could be exploited using human photoreceptors that people will be transplanting in the clinic. And maybe we could make those transplants more successful in terms of helping people see better after the procedure.
I want to bring in our guest scientists, our rising stars, but I know there's a few others you want to speak to who aren't with us today. Take it away in terms of, first of all, Dr. Philippe Monnier.
Yeah, so we can't highlight everybody today out of the interest of time, but I'd just like to emphasize that, the group has a lot of people working on how to improve retina function in disease. So Dr. Monnier has been studying proteins that help neurons connect to where they make their connections in the brain. And what he's shown is that this particular set of molecules that help make these connections act way more broadly than what people initially thought. And more recently, he's shown that these proteins are involved in the death pathway of photoreceptors in inherited diseases. And these diseases model what happens in people. So by targeting these proteins, he's been able to delay the death or the degeneration of photoreceptors. And that's exciting because it looks like it works in different contexts, different types of diseases. So maybe this could be deployed as a more general strategy to help slow the death of photoreceptors in many kinds of photoreceptor diseases like retinitis pigmentosa, but also macular degeneration. And our other investigator, who's not joining us tonight, but I'm sure you'll meet him in other forums like this, is Jeremy Sivak. So he's working on the neurons that cause the problems in glaucoma. So I've talked a lot about photoreceptors. There's a whole other disease, age onset disease, that's very difficult to treat, and that's glaucoma, and that's the death and degeneration of ganglion cells. Those are the cells that connect the eye to the rest of the brain. Without those cells, you also will lose vision. And it's been very hard to kind of rescue those cells in times when they're traumatized or stressed. And he's identified chemical messengers that are made by cells that surround those ganglion cells that seem to help those cells survive chronic stress or injuries. And, you know, that's potentially a very interesting set of chemicals to target that could eventually be deployed into strategies for long-term protection of neurons that might die in diseases like glaucoma.
Thanks so much, Dr. Wallace. And I want to ask you to stick around if you can till the end of the hour. We'll bring you back in with Dr. Weaver. A quick reminder to our audience, if you have a question for our scientists, we invite you to click on the link at the bottom of your screen or go to slido.com and type in code. So that's the actual word CODE 5875. You can submit and vote up questions. I'm told already that we do have a number of questions coming in. So if we need to, I'm hoping people can stick around a little later after the bottom of the hour in case, so we can get to as many questions as possible. All right, let's turn to our rising research stars, all recent or fairly recent recruits, and all cross-appointed to both the Donald K. Johnson Eye Institute and the Krembil Research Institute. First up, Dr. Michael Reber. He's a senior scientist at the Donald K. Johnson Eye Institute and the Krembil Research Institute. Dr. Reber is a world-renowned neurobiologist whose research focus is restoring vision by retraining the brain using virtual reality. In 2017, Dr. Reber was recruited from Strasbourg, France to UHN, a move that was actually facilitated and made possible thanks to donor support. Welcome, Dr. Reber.
Hello, Christian. Good evening, everyone.
Now, this is really fascinating to me, being a visual person myself. You're pioneering the use of virtual reality as a treatment for patients with visual impairment to help them restore vision. Let's sort of frame the narrative first, Michael, if you don't mind, with visual impairments. What are we talking about here, and what's the burden of this impairment in Canada?
So, I mean, the visual impairment we are talking about here are the ones that cannot yet be cured. The ones Dr. Wallace was just mentioning, such as macular degeneration, glaucoma, or visual field loss consecutive to stroke, trauma, or cancer. And what is striking, and to me, actually, when I arrived in Canada, is that, for example, every 12 minutes, someone is diagnosed with visual impairment in Canada. And there are more than 5 million Canadians affected with visual impairment. And when you know that visual impairment increases your chances of depression, for example, the chances of depression are multiplied by 3 when you have vision loss. All those numbers were just, to me, quite stunning. And I must say, even worse, I would say, for 70% of Canadians, the most feared disability is vision loss. So this tells you everything, I would say. So these are definitely strong impact on quality of life.
And what's on offer in terms of treatment?
Well, right now, I mean, and the ophthalmologist could also answer that question. But for example, for macular degeneration, you would have anti-VEGF injections in your eye. For glaucoma, you would take molecules that would decrease the intraocular pressure. For field loss, for example, you would use prisms on your glass that would displace the missing field or the blind field. So these are currently the treatment available, but they can slow down the disease, but none of them will actually cure. So once the visual impairment or the visual loss is there, it's there.
And so we'll turn to your research and this is what's fascinating for me is number one, your research is based on something that's not invasive, and second, it's based on a principle. You're leveraging a principle, right, about the plasticity of the brain, its ability to rewire itself. So talk to us about this, how virtual reality lends itself to this rewiring of the brain principle.
So indeed, we use the one of the principal feature of the brain, which is plasticity. This means that actually the brain is capable of reorganizing or rewiring depending on the stimulation. For example, each time you learn something new, your brain rewires partially. So the learning induces an adaptation of your brain, and this happens whether you learn a new poem, you learn playing an instrument or a new sports, and this happens at any age, whether you're young or adult or even elderly. So for sure, I mean it's faster in young person, but elderly can also learn those things. So basically, we are teaching the visual system to somehow maximize the use of the healthy cells left, and for this we use virtual reality.
All right, so let's walk through the process. You are, first of all, you are trying or investigating with real patients and applying a particular kind of training using the virtual reality headset. So walk us through what you have the patients doing.
So actually, that's the big news, because we had two recent manuscripts that got accepted for publication, one last month and a recent one last week. So we are very happy with this. So basically what happens is that those patients, and for now we've been working with patients with visual field loss consecutive to brain cancer. So those patients are unable to see half of their visual field. And what we do is we provide them the VR, which is the thing which is sitting here behind me.
Right.
It looks like this. So you would wear that and then perform specific visual tasks resembling a little bit the second one that I have below here with spheres moving, and this stimulates your vision and your visual system and your visual network. And this basically, as I said, somehow pushes and maximizes the healthy cells or the healthy connections that are left. And so the patients basically perform those stimulation or visual stimulation tasks repeatedly for now. Our protocol runs for 6 weeks. So basically every 2 days for 6 weeks, for basically 15 minutes a day, the patient perform those visual tasks. But the beauty of it is that this is all performed at home. So the patient don't have to come to the clinic. And that's, I believe, is a major improvement, I would say, especially when you have low vision or impaired vision. You don't want to commute and go around and spend every other day going to the hospital. But you can do this from home, from your home, and we control the device with our computers in the lab. So we exactly know what the patients perform, what type of visual stimulation they perform.
And what can you tell us, Dr. Eber, in terms of results thus far?
So far, I mean, we've seen in those patients with field defects, we've seen a dramatic improvement in metrics, ophthalmologic metrics, such as contrast sensitivity, such as fixation stability. Fixation stability is very important when you want to read. You must be able to fixate your gaze, otherwise you can't read. So we improve fixation stability in those patients, but we also, of course, improve field restoration. So these patients are now perceiving visual stimuli in what was corresponding to the blind field previously. And of course, this all impact the quality of life, which dramatically increased in those patients.
So, talk to us about where you intend to take this in the future.
So the future now is really to somehow design specific visual tasks for specific eye disease. So one can imagine that if you have macular degeneration, you might perform different visual tasks than if you have field loss, for example. So that's where we are now, and thanks to the support of the donors and the Krembil Foundation, we are able to now start very soon a clinical study on patients with macular degeneration, and so we will test this device on patients with macular degeneration. But also the work that was done previously was also possible because of the support of the donors. So the two recent publications were made thanks to the support of the donor to start those feasibility or so-called pilot studies.
Well, continued success to you. And one more question for you. You're a well-established scientist from France. You had a successful career going at the Institute of Cellular and Integrative Neuroscience in Strasbourg. And then a few years ago, you decide you want to uproot your life and your career and join the Donald K. Johnson Eye Institute. What was behind that move?
That's a good question. I like challenges, but no, the thing is, I knew that the Donald K. Johnson Eye Institute and the Krembil were really connected and integrated in the hospital. And I must say, when we perform research like we do, combining both basic but also clinical research, you have to be close to the clinicians. And the fact that the Donald K. Johnson Eye Institute is integrated, I would say, in the Toronto Western Hospital, right? The buildings are right next to one another. Mm-hmm. And that within that Eye Institute, both clinicians and basic scientists work together. This facilitates all type of collaboration, and especially when you want to translate the research from basic to clinical. And this is, I must say, something which actually does not occur that often, and you don't see it so much, and not in such an efficient way. So where I was in Strasbourg, this wasn't existing, and that was some sort of a hampering my research and my objectives on the long term. So that's one of the reasons I came. The other is also all the resources that are available here and the expertise of my colleague scientists, but also clinicians. So I think the decision wasn't too difficult to make. And as I said, I like challenges and I like going out of my comfort zone.
Well, a belated welcome to you. Next up, Dr. David Matthew. He's a clinical fellow from Kerala in southern India. And David is a surgeon at the Toronto Western Hospital, as well as he's training to become a research scientist. And he's focused on investigating new treatments for glaucoma. Welcome, Dr. Matthew.
Thank you. Happy to be here.
Help us first understand, Dr. Matthew, what is glaucoma?
Right. So glaucoma is a group of diseases that affect the optic nerve. That's the nerve that connects the eye to the brain. It's sort of like a wire between the eye and the brain. In glaucoma, the most characteristic feature would be increase in eye pressures. To expand on that a bit, eye pressures created by the fluid that's in the eye. So just imagine a balloon that won't expand in size, but the pressure inside goes up. That's something that happens in the eye when the pressure goes up, and that increased pressure in the eye causes damage to the nerves. And this damage to the nerve affects the vision, which progressively gets worse till the patient becomes really blind. That would be the end stage of the disease. So there are different types of glaucoma based on the structure of the eye and how the drainage works. Yeah, that's in short what glaucoma is.
Thank you. And I understand, of course, much of research for those treatments uses animal models, in one of the most common being rats. And I understand, David, a few years ago, you actually made a striking observation about those animal models that were being used in glaucoma research, and that there was actually kind of a weakness in the models they were using. What was it you discovered?
So to study any disease, we need models, animal models. So the most commonly used ones are rat and mice models in glaucoma. And what we do with models is that we artificially induce high pressures in the eye to mimic glaucoma, so we can study the disease. In all created or artificially induced glaucoma models, the pattern of increase in pressure is such that there's a huge increase in pressure to close to 50 to 70 or even higher at the creation of the model, and then it slowly comes down over time. But this is not what we typically see in most glaucoma patients. We see a gradual increase in pressures rather than a marked increase in pressure as we have in most models that are existing right now. So that I found was a big weakness to start off with. We need a model that resembles clinical disease to study the disease.
Exactly. I mean, to me, that's fascinating that you were able to figure that out, which is so basic and perhaps was not helping glaucoma research. So you essentially set out to create a better mousetrap, or in this case, animal model in terms of the rat, that would give all glaucoma researchers a more accurate method of studying the disease. Walk us through what you did.
Right. So first off, I'd say that creating a new model is difficult. It's a really challenging job. And there was an already existing model that used a suture material, or a sort of a small string that was passed around the eye and tightened so that it squeezed the eyeball and created high pressure in the eye. I used a modification of that technique. Instead of tightening it up, I left it loose so that there was no squeezing on the eye, and the pressure was sort of close to normal at creation. And over time, the suture gradually tightened, and that led to increase in pressures slowly over time. And this created a model that we were trying to create. And yeah, and that's how I sort of modified this already existing model.
And another area of your research is discovering the potential benefit of a molecule called lipoxins that might actually perhaps help in the prevention of glaucoma. Talk to us about lipoxins and how you connected their potential for helping patients with the disease.
Yep. I'm a part of Dr. Jeremy Sivak's lab, where I'm doing a PhD. So Dr. Jeremy Sivak and his excellent researchers have already are quite interested in this a class of molecules called lipoxins. These are small fat molecules that are produced within the body in response to different tissue injuries and inflammation. It's already been studied in the context of other diseases in the body, such as diabetic kidney disease and metabolic syndrome, and even Alzheimer's disease. And in all these diseases, it's been found that the levels are low in a diseased condition. And in preliminary studies on cells in a dish, our lab has found that this is the case even with a nerve injury, or specifically a glaucoma injury. And a supplementation of these molecules did prevent increasing injury in these models. So we have used cells as well as some acute animal models to study this. Next step is to use our improved model to study this further in an animal model.
Amazing. You're a clinician scientist, originally from South India, as we mentioned. Talk to us about your journey here and what motivated you to join the Donald K. Johnson Eye Institute.
Yeah, so I came here in 2017 for a clinical fellowship in glaucoma. So after 1 year of training in glaucoma management and surgical procedures, I decided to spend a year in Jeremy Sivak's lab. And this was made possible by generous donor support, and I'm thankful for that. And during this year, I came to know about different challenges in the field, and especially in research in glaucoma. And that's when we talked about the model and how we started the work. So in the past, I've always wanted to do research, but in India, I quite did not get around to doing it much because research is always swamped by clinical work. And here, there is protected research time, and that is very important for clinician scientists.
Terrific. Well, a belated welcome to you as well, Dr. Matthew. Next up, Dr. Karun Singh. He's a senior scientist at the Krembil Research Institute. Dr. Singh's research focuses studying the trajectory of eye disease using stem cells with the goal of developing drugs to halt the disease. Thanks to donor support, Dr. Singh was recruited to UHN just last year in 2020 from McMaster University's Stem Cell and Cancer Research Institute, where he was the neural program lead. Welcome, Dr. Singh.
Thank you very much, and welcome everybody.
Okay, Karun, let me see if I have the basic premise of your work down, and interrupt me at any point, because I think it's really a really cool new research path. You're leveraging the potential of stem cells, but in this case, not for therapy or for regeneration, but for study, so that you take the stem cells, grow them into eye disease, and examine the progression from A to Z of the eye disease. And then the goal is to generate thousands of combinations of molecules, which would become a drug essentially, to try and come up with that drug that will slow down or reverse eye disease. And you can actually try out all these combinations on the eye disease that you've grown. And the really cool thing is, this is human tissue that you're working with in a petri dish. Have I got this right?
You absolutely do. You do. That was spot on.
Okay. Well, thank you for coaching me. So let's examine this process, because it all starts with taking a simple blood sample from a patient with eye disease. What happens from that point on?
Yeah, so this is really amazing, very exciting technology where we can actually grab a simple blood draw, one tube of blood that you would normally, go to get blood work done. That same tube of blood we can grab, and from that one sample, we are able to use genetic reprogramming, and so we can actually genetically engineer your white blood cells to become stem cells in the dish. So now we have this infinite source of stem cells that are in a dish, and those cells can become any cell in the body. And what's really cool about this technology is that we can actually use those stem cells to make eye tissue in a dish. So this is really, really important, because I myself in my own lab have been using animal models of disease, brain disorders for the longest time. And one of the issues is that while using mice or other animal models are important, it's always not exactly human tissue. And so really we need to have, and getting human tissue, whether it's brain samples or even eye tissue, it's just not ethically feasible to do that in a live person. So we have to come to a compromise. And so this technology really helps us get there. So with these stem cells that are generated from a specific person with eye disease, we can make their eye tissue in the dish, and we can study their disease and try to treat it with different combinations of drugs. So it's very powerful.
And you end up, and again, I'm trying to go on memory here in terms of this progression of the blood sample, stem cells, coaxing them to stem cells, and you get them to progress into eye disease. You end up with something called a 3D retinal organoid. Can you explain what that is and the significance of it?
Yes. So in the past, what we were normally using or making actually are stem cells in 2 dimensions, so flat cells. And then we would make those into 2-dimensional eye cells or brain cells, for example. And so while that's amazing to be able to generate human cells in the dish, they really weren't that mature, and they weren't really representing what's happening in the actual eye. So now in 3 dimensions, we can grow what we call retinal organoids. So these are 3-dimensional tissues that are from a person, and we did not have to go to their eye to get this. We generated them from just a single sample of their blood. And what is really exciting about this is that those cells are very mature, much more mature, closer to their eye than would be 2-dimensional cells. So that's why we call them retinal organoids. It's a very new technology, just a few years old. It's very fast developing, and it's exciting because for myself, who used to work on animal models, this is to me really the next wave of future research endeavors that a lot of people are kind of being drawn towards, because it's human. We did not really have to go and impact patient life very much. We just needed a single tube of blood to be able to make their own version of eye disease in the dish. So it's very exciting.
So talk to us then, Karun, next steps. Like, where are you at, say, on the therapeutic front in terms of these combinations of molecules and testing them on the samples in the dish?
Yeah, so right now what we're able to do is make different types of eye cells in the dish and in 3 dimensions. One of the main things that we're trying to do is scale this up. You can do this in a very boutique fashion from a few patients, but what we really want to do is be able to scale it up to the point where we can screen hundreds or thousands of drugs on an individual's cells or their 3-dimensional retinal organoids that represent a specific disease. And so by doing that, we can screen thousands of drugs to try to find the one or few that might work in their disorder. So this is really exciting because it's really getting more towards personalized medicine, where we can kind of tailor the therapy for the individual, very similar to what the cancer field has been able to do and achieve, where you have a specific form of cancer and you have a specific medicine that might work for that. This is exactly what we're trying to do in the retinal field.
And is there any opportunity, say, for cross-pollination of applying what you're learning when it comes to vision to other parts of the brain?
Yes, exactly. So actually part of my lab actually has been working on various brain disorders. So things like Alzheimer's and Parkinson's. This is a huge advancement in being able to model and study a patient's own brain disease in a dish, because obviously biopsies are not possible. MRI is very important, but it's really a 10,000-foot view of what's happening in the brain. We need to be able to get at the nuts and bolts of why disease is occurring, detect it early, and then try to treat it with pharmaceuticals or different drugs. And so for me, the Krembil Research Institute allows me to kind of work both in the retinal field and in the brain field. And so we're actually working with lots of different researchers here on Alzheimer's and Parkinson's as well.
Wow. Okay. Listen, talk to us about your motivation for being here. You joined the Donald K. Johnson Eye Institute just a year ago. You're just down the road in Hamilton. What motivated you to pick up your entire lab, I understand, and bring everyone down to the Krembil?
Yeah, so it was a big decision, but it was really the right decision for me. I've always been very excited. I've trained in Toronto in the past to do my PhD, and I've always known that Toronto is the place where cutting edge neuroscience is occurring. And what I know about the Krembil Research Institute before and what it is now is completely transformed. It is such an exciting place. For someone like me to really make a mark in research, I need to be very close to clinicians. I need to have access to patient samples. And really, it's more of a collaboration between the scientist, the clinician, and the patient. So that really is the perfect storm of research, and that occurs here at the Donald K. Johnson Eye Institute. So for me, it's through the donor support, it's been absolutely tremendous. This last year has been so exciting. We've launched just a number of new research projects in the last year that I never would have dreamt would be possible, and that's all because of being here in Toronto.
Well, welcome to you, Dr. Singh.
Thank you.
And finally, Dr. Brian Ballios. He's the latest recruit to the Donald K. Johnson Eye Institute. In fact, he is yet to join us. I understand that he's moving day is later this week. Dr. Ballios is a clinician scientist specializing in treating inherited retinal disease with a research focus in cell therapy. And again, it's thanks to generous donor support that Dr. Ballios joins the Donald K. Johnson Eye Institute. He's currently completing a clinical and research fellowship through Harvard Medical School's Training Hospital, Massachusetts Eye and Ear in Boston. Welcome, Dr. Ballios.
Hi, Christian. How are you?
Great to have you here and having you join the Institute. Your research focus is developing cell therapy for transplantation to treat blindness. Let's start with, to your mind, what's at stake here for patients?
Well, the patients and the conditions that I'm particularly focused on treating are ocular genetic and inherited ocular conditions. And these are conditions for which there really is no standard of care. These are conditions that are generally progressive, and they're often conditions, almost invariably conditions that affect multiple family members or run in families. They have significant disease burden for these patients, and these are some of the patients that are probably going to be the most challenging in ophthalmology to treat, since we have no current therapies. And these are ones for which the next generation of therapies to treat retinal disease are going to be focused on having impact.
So you implant stem cells into the damaged retina to help regrow light sensing cells and restore vision. Talk to us about what you've discovered about what makes a viable cell to send in and where you're at on this front.
Well, one of the exciting things is that stem cells and cells that stem cells make have already entered clinical studies for patients with these conditions. And what we're learning and what we're understanding is that whereas early we thought that all these conditions, inherited conditions, would be amenable to stem cell transplantation, we're understanding that the stem cells may behave very differently in different disease conditions. For inherited retinal disease, there's over 250, almost 300 genes that when they have misspellings in the DNA can cause these retinal conditions. And we're understanding now that that cells that are transplanted to try to regenerate vision and to replace the light-sensitive cells may actually behave very differently depending on which specific gene is causing this retinal condition. And so one of the goals of my research program is trying to understand, well, what's unique about each of these conditions? So what's unique? Are there some general principles that we can pull out that will help to improve the efficiency and the efficacy of these cell transplantations in disease?
And I understand another challenge in research when you're doing this kind of transplantation is the actual method of delivery. Talk to us about, you've actually pioneered a method that really ensures that the stem cells get to where they're supposed to go, right?
Yeah, so when I was training in Toronto as part of my research, and our group was the first to develop an injectable biomaterial-based delivery vehicle to deliver stem cells to the retina, resulting in viability that had never been seen before supporting these cells after transplantation. And our group continues to build on that work. And so this speaks to how we have to think about new and innovative ways to get these cells to the places they need to be and support them while they do the business of regenerating vision. And whether that be through the material transfer phenomena and approach that Dr. Valerie Wallace touched on in her talk, whether this be through making connections with the next cells in line in the retina. Gene therapy as a parallel field has already shown us that if we can regenerate light sensitivity to light sensitive cells, that the rest of the retina is ready to receive it. And there's now an approved gene therapy that is now approved by Health Canada, waiting to be applied to patients. So we're so encouraged by the work in those parallel fields that we're certain that in the coming years we'll see new therapies developed for patients with genetic eye disease.
And Dr. Ballios, what can you tell us in terms of, tell us about what the various trials you have ongoing? What kind of results are you seeing so far?
Yeah, so I'm involved in several trials right now involving gene therapies and also stem cell therapies. And what we're seeing is an excellent safety profile for the cell therapies that are being used in the retina when they've been thought of carefully. And one thing that we're looking for and continue to look for in the trials is moving to patients with better and better vision. Now that we have the benchmark of safety of this living therapeutic in the retina, now we're understanding what effect is this having on vision and the important outcome measures of vision, visual acuity, field of vision, light sensitivity in these retinas. And we're trying to understand that better and better. And we're also understanding that maybe there's other types of cells we should be thinking about transplanting. Maybe we should be focused on some of the very specific light sensitive cells that we can make from stem cells to treat specific human retinal disease. And those are the sort of discoveries that are coming out of this work right now. So it's a time for patient engagement, and it's a time for discovery in this field.
So your lab, paint us a picture of where you're going to be at in 5 years, say, once you join the Institute.
Well, so for my particular research program, as I touched on before, I'm very interested in understanding specific human disease and how these genetic and inherited retinal conditions manifest, and how stem cell therapies may behave differently in the context of these different human diseases. And so my research program touches and synergizes with a number of the programs you've already heard from this evening, understanding modeling human disease, the specific inherited conditions, and how do stem cells potentially interface with these conditions differently. And so I anticipate in the coming years, as we understand more about these stem cells, we'll be starting to think of designing, let's call it cell therapy 2.0 for inherited retinal disease, something more specific, and thinking about designing the next generation of trials to study cell and light sensitive cell transplantation.
And what's motivating you to join the Donald K. Johnson Eye Institute?
Well, I trained and grew up in Toronto, but it really coming to UHN is about joining an ecosystem of discovery. I mean, we have the best of the best clinicians and vision science researchers, and we have researchers and clinicians who work hard and they care hard for patients, and that's the kind of group that I'm really excited to be part of.
Well, we wish you well and welcome and have a safe trip back on your move.
Thanks.
All right, before we get to our audience questions, I want to quickly bring back in Drs. Wallace and Dr. Weaver. I have to say, this lineup of scientists you've been busy recruiting is just so impressive. What does it say about the Donald K. Johnson Eye Institute?
It says that the future's bright. It says that Valerie has good vision.
Oh, Don.
I'm sorry. I also wanted to work in that our fundraising should have been last year because 2020 was the best year for fundraising. I'll let that one go. What it says is that, as I said, the future is good. You build upon young minds, and we have fertile young minds who are going to be bringing us the discoveries of tomorrow.
Okay, the fundraising issue, because it's important to bring up, because something I've learned in my years here at UHN is that research in Canada has kind of this catch-22 ongoing every day. It takes time, it takes a lot of money, and yet there is no base funding for research, medical research in Canada. I looked up, I was talking with Brad Waters, the EVP of Science and Research at UHN, and he mentioned the fact that in 2020, UHN Research had over 700 different groups to fund research projects, and they're often all tied to specific projects. So you can't go to the Ministry of Health or UHN for funding. You are constantly chasing money. So talk to us, you two, about the key role that philanthropy plays in this very challenging model, this financing model across Canada, actually, for medical research.
Valerie, you'd be happy to address that issue.
Oh, I'll be happy to do that. So if you think of a research organization as a big house, we may go, let's say, to CIHR and health charities to, get groceries. That's what pays for the actual, making experiments, the reagents you need for experiments. But that's not the whole house, and so funding the house is much more difficult. And also, you can have a small house, or you can have a house with like a hot tub and, a wine cellar. And I'm not saying we've built wine cellars at UHN, but when you have that kind of infrastructure, like gourmet kitchen, for example, you're going to do better experiments. So how do we get the better features in the house? And that's through philanthropy. That it would not be possible. So, this is a great group of scientists. It's a very exciting time. But they would never come here if there wasn't support for them and if there weren't resources. So, they need a gourmet kitchen. So I hope they would agree that they have some elements of a gourmet kitchen to work in. But all those things are possible because of philanthropy. There's very few other places where you can get that kind of funding. So it's just making a great, much better house. And there you're going to make better experiments, or you have the opportunity to make great experiments.
Well, and all four of these people look like amazing chefs. So best of luck. All right. We are really pressed for time, but we need to get to our audience questions that are coming in. Let's bring in our Slido slide, and everybody jump in with who should be appropriately answering. First off, how close are we to seeing a future with the possibility of retinal regeneration? Will those with severely impacted retinas have the chance of seeing again? I'm going to go to Dr. Ballios.
Great. So I think we're living in that future right now. I mean, we spoke quite a bit about cell therapy. I think cell therapy is learning from the success that's already being experienced with gene therapy. And so we're seeing retinal regeneration of vision. We're seeing improvements in night vision. We're seeing improvements in side vision. We're seeing improvements in retinal sensitivity and visual acuity in patients already who have conditions which never had treatments before. And so I think we're living in that future right now. And what we're thinking about is really growing the menu for patients, using that gourmet chef analogy. We want to give patients options. We want to give them the potential to choose drugs, to preserve their retina for longer, to give them backlight sensitive cells, to make those cells they have work harder. So I think that we are living in that future, and we're moving to future 2.0 soon.
Next question from Jane Burfield. Is research leading towards a different form of sight, like cochlear implants have led to hearing? Who is going to answer that one? Brian?
I can take that again if you like.
Yeah, Brian, I think that's good.
I'll hop on that one. So this is a fascinating question. So there are other forms of sight that we don't yet appreciate, and there is research into understanding the clinical application of that. A good example is something called optogenetics, which is using gene therapy to put a turn a light sensitive cell, or a cell in the retina that's not normally light sensitive, into a light sensitive cell. And that's going to lead to a different form of sight. It's not using the same light sensitive cells that you were born with. It's making cells that aren't normally light sensitive. So what does that vision look like? And how will patients need to train to use that new vision? This is going to touch on the great work that Dr. Reaver's doing. And so there's opportunity in the future for different forms of sight, certainly, than what we're used to thinking about.
Next question from John. Recommendations for eye sun protection, factors to consider: fit over versus clip-on lens, or clip-on lens quality, UV, A and B filtering, polarization, tint, color, blue light. Who might want to take that one? I'm going to say Brian.
Brian, yeah.
There's so many clinical questions. I'm happy to answer. Patients ask me this all the time.
Lucky you're coming here.
That's great. So patients ask me this all the time, and I say, well, first off is comfort. But for some conditions, we also think about like macular diseases, age-related macular degeneration, or inherited retinal conditions. I usually recommend something that protects against UV light. It doesn't matter what particular brand of sunglasses or particular shade or tint, but something that protects against UV light. There are tints that may confer some more comfort for patients with certain eye conditions. We don't have time to go into that. Blue light filtering is something which some patients report having more comfort with their vision and less eye strain when using the computer. We don't really have good evidence to understand the effect of blue light filtering, but I think definitely UV light protection for a number of different eye conditions is important.
Appreciate it. Thank you, Dr. Ballios. Next question from Eileen. Can peripheral vision lost by brain surgery be restored? Dr. Ballios.
Yeah, this might also be a good question for Michael. In terms of his training of the brain and the peripheral field. I think that we don't yet understand the plasticity of vision and what we can be retrained to see when we lose parts of our peripheral field. And this starts to touch on the concepts of vision rehabilitation, which I think is such an important part of therapy for all the different kinds of retinal and ocular conditions that we've been discussing today. So I think that we can improve parts of our peripheral vision, but I think also we have the potential to regenerate peripheral vision in the future with some of the therapies we've been talking about.
Dr. Reaver, you want to jump in?
Yeah, sure. To specifically answer that question, I mean, first you would want to identify which brain areas were affected by this surgery. And then if those were identified using imaging techniques such as fMRI or so, then we can somehow elaborate on a very specific visual stimulation procedure, which would, for example, bypass that area or try to stimulate still some remaining cells in that area. So, but yeah, that's all complementary to what Dr. Ballios just said. So yeah, this goes all together.
All right, next question. What is the current state of the research for a cure for chronic dry eye disease? Who can I turn to for that one?
I think Dr. Ballios probably
I'll put my hand up again. Yeah, so this is getting a little further to a part of the eye that I don't spend as much time focused on, but chronic dry eye is a significant burden of disease for many patients, and there's a significant amount of research going into understanding what exactly causes chronic dry eye. Is it immune system mediated? And what are the factors that are either inflammatory or otherwise that go into causing dry eye disease? And with the variety of medications on the market right now for either lubricating the eye or anti-inflammatories or immunosuppressants, trying to understand how these drugs interface. So I think that what we'll see is just further work in this field for dry eye disease. There are some specific types of chronic dry eye that are the result of an absence of stem cells on the surface of the eye. There are many researchers looking at understanding whether encouraging the growth and development of these stem cells may ultimately help with chronic dry eye in a more permanent way.
Next question. Is there any research being done on regenerating retinas in those whose issues are related to trauma and not age?
I think I might be able to speak to this. We're interested in, so trauma, for example, if a big part of the retina is damaged, I could imagine that the cell-based therapies we're interested in or working on could one day be applied to a case of, restoring vision after trauma. We tend to work on very predictable models of degeneration because we know them so well and they're well characterized. But I don't see why in the future for issues of non-kind of chronic disease causes of vision loss, why some of these approaches wouldn't be amenable to those kind of treatments or for those kind of conditions.
All right, thank you, Dr. Wallace. Next question. Are these treatments presented today available for people who want to volunteer? That's a great question, actually, for people wanting to enter trials.
I think, that is all very context specific and it depends on the trial. And so there's usually very, very specific criteria for enrollment. So it's not like anybody, it's for issues of safety, for the design of the trial that patients are selected very, very carefully. And so it's not a case of, just randomly selecting people. When there is a trial that deals with a certain condition, then the word goes out that there's enrollment taking place. And I think Brian's been involved in quite a bit of that in Boston, and he might have more specifics to add to that.
I would only add the comment that, this is really important. Patients come and ask, what I want to be part of when can I receive this treatment as part of a trial? And I like to emphasize that clinical trials are not treatments. They're trials. They're about trying something and they're really about an experiment and trying to understand, is that new treatment going to be safe? Is it going to be efficacious? Which patients is it going to be best in? And so I think that's an important thing to understand. And so the decision to participate in a trial or a study ultimately becomes a very personal question for the patient. It's about a risk-benefit analysis. It's about understanding that it's completely voluntary. And it's also about understanding that it's okay not to participate in a trial. It's also okay to make the decision to wait for an approved treatment or therapy. And so patients, I try to also encourage them to feel that they're not missing out on the advances that are going on if they should choose for their personal reasons not to participate in a trial. So I just add that point.
Much appreciated. And look, I appreciate all of you for staying on after our allotted time. I appreciate you telling us your stories and your latest work. It's fascinating. That's going to have to be our final question tonight because we've gone past our time. I want to thank all our guests today for giving up their time and sharing their stories. Dr. Donald Weaver, Dr. Valerie Wallace, Dr. Michael Reber, Dr. Karun Singh, Dr. David Matthew, and Dr. Brian Ballios. And to our friends out there in the audience, thank you for joining us today, all the Discovery Ball supporters, and a big thank you from UHN Foundation for your continued support. For a recording of today's session, and of course, if you feel so moved to be able to donate or give, keep an eye on your email inbox. Thanks for tuning in, everyone. Stay safe.